Okay, the title of my talk today is Red Meat and Heterocyclic Amine Intake, Metabolic Pathway Genes and Bladder Cancer Risk. In this study, we test the hypothesis that high meat intake is associated with increased risk of bladder cancer. And I would like to give you some background information on bladder cancer. Bladder cancer is the fourth most common cancer in men in U.S. In 2009, about 70,000 new cases were diagnosed with bladder cancer and with 14,000 deaths. The rationale behind our study is that meat cooked under high temperature generate heterocyclic amines, which is called HCAs, are carcinogens that can cause cancer. And after the consumption of HCA into the body, HCA interact with metabolic enzymes, and these enzymes bioactivate HCA and then detoxify HCA. And because the level of the enzymes are genetically determined, so people with different genetic background in the metabolic pathways of HCA may be at different risk of cancer. So in this study, we ask two questions. The first is there an association between meat HCA intake and bladder cancer risk? And would the genetic variants in the HCA metabolic pathway modify the association? We did a case control study. Our population is based on a Texas bladder cancer case control study led by Dr. Xifeng Wu, who is also sitting in this room. Our bladder cancer cases are newly diagnosed cases recruited from MD Anderson Cancer Center and Baylor College of Medicine. They are previously untreated and histologically confirmed. There is no age, gender, ethnicity, or stage restrictions on recruitment. Our controls are recruited in collaboration with Kelsey-Seybold Clinics, which is Houston's largest multi-specialty medical organization. Controls have no prior history of cancer, and controls were frequently matched to cases on age, gender, and ethnicity. After informed consent, all study participants completed a 45-minute epidemiological questionnaire and a 45-minute food frequency questionnaire. In the food frequency questionnaire, frequency and the portion size of each meat item were queried, and also cooking methods and the doneness level of each meat item were queried with color picture to standardize the response. And then we did a comprehensive literature review to select genetic variants involved in the HCA pathways. We also used bioinformatic tools to refine the gene list. And then DNA were extracted from blood samples from all participants and perform genotyping. In total, 217 genetic variants in the carcinogen metabolic pathways were genotyped. Here are some of the results. Compared with controls, cases consumed significantly higher total meat intake, processed meat, and red meat intake. And in terms of bladder cancer risk, we found no association between total meat intake and bladder cancer. But interestingly, for red meat intake, compared to the people with lowest level of red meat consumption, those at the highest level of red meat consumption is at 17-point-fold increased risk, and there is a significant dose-response trend. So the more red meat a person consumes, the more increased bladder cancer risk. There is no association between processed meat, poultry, and fish intake with bladder cancer risk. And then we analyzed individual meat items that associated with cancer risk. And these are the list of individual meat items that had significant association. HCA, we calculate HCA levels in meat items, and then calculate the total HCA. So as we go from the lowest HCA intake to the highest, bladder cancer risk increased significantly with a dose-response trend. doneness level of meats compared to the rare done meat, the medium done level is associated with 1.4-fold increased risk, and the well done and very well done meat, the risk increased to almost twofold. This table shows the joint effects of red meat intake and the metabolic pathway gene on bladder cancer risk. In this table, the rows are sorted in order of lowest red meat intake to the highest, and the columns are sorted into 3 categories of unfavorable genotypes, a number of unfavorable genotypes. So within each row, we can see that within each row, which is in a certain level of red meat intake, bladder cancer risk increased with the number of unfavorable genotypes. And within each column, that means with a certain number of unfavorable genotypes, bladder cancer risk increased with the level of red meat intake. So our reference group is the number of people who had lowest level of red meat intake and who carried the lowest number of unfavorable genotypes. As we can see, the highest risk group are people who consumed the highest level of red meat and who carry the most number of unfavorable genotypes. We can see that the risk is almost 5-fold. So this is a kind of people who are the most susceptible in our study population. In conclusion, our data show that high red meat intake was associated with increased risk of bladder cancer, and consumption of heterocyclic amines generated by cooking meat in high temperature increased bladder cancer risk in the study population. And our data also show that red meat intake and bladder cancer association may be modified by genetic variants in the carcinogen metabolic pathways. Thank you.